Okay, now that I've talked a little bit about how RNA is made during the transcription process, we're going to talk for a minute just about how it's edited. So the RNA that comes out of the process, out of the RNA polymerase, sort of shown up here, this red line, it is not used in this form. It needs to be edited, and I'm going to talk for a few minutes about what happens in that process. It's almost like if you write a paper, you don't write down, you don't turn in the first draft that you've written. You have to go back and edit it and get rid of some parts and add others. So that's what we're going to be doing. So the newly transcribed strand is called pre-RNA. It needs further processing in order to be useful. It has some non-coding sections, and these are called introns here. And the coding sections of it are called exons. So in any given strand of mRNA, there could be quite a bit of it that is not coding for proteins and needs to be edited out before it goes into the translation process. So we've got to cut these things out, and then we're going to splice the exons, what the coding part is, back together. It's pretty simple. So if you look at this diagram, here is a DNA. All right, here's our DNA molecule, and you see that these exons are in these colored blobs. And those represent coding sections. And we might think of this entire sequence as a gene, and the gene can contain some exons, which code for proteins, and introns, which do not. So after the mRNA is transcribed, it goes through this editing process. The introns are removed, and then the exons are spliced together. It's called RNA splicing. And then you end up with your edited copy of the RNA. And here's another look at this. Here's our DNA here, and it shows the exons and the introns, and then it's transcribed, and in the transcription it includes the introns, even though they're not used, and then there's further processing to turn it into mRNA, where the introns are removed, and it can be used to translate proteins. Now I want to just show you this here. This is 5' cap and a poly A tail. I'm going to talk about that next. So further processing, the 5' cap. So a methyl group, and that a methyl group is just CH3, carbon and 3 hydrogens. That's a methyl group. That's added to the 5' end of the new mRNA molecule. And the purpose of this is just to help the ribosome recognize that 5' end during the translation process. So you've just got here, you can see it here, it's just got this little cap on here. It's called a methylated cap. And then in addition, on the other end, on the 3' end, there's a string of 100 to 300 different adenine molecules, and they're just a bunch of A's, and these are added on to the very end, and that's called a poly A tail. And really the purpose of this is to protect the mRNA. It's going to go out into the cytoplasm. There's enzymes and things out there that want to destroy it, and the process is complex, but basically having this tail on here acts as a bit of a shield and protects the mRNA from those enzymes. So that's our processing, and once we're done with this, it moves through holes in the nuclear membrane. So this green represents the nuclear membrane, and there's these pores in there. Nuclear pores are just holes in the membrane. You might remember membrane structure for biology, but we won't go into it here. In any case, the mRNA is going to leave the nucleus and go out into the cytoplasm. You can see the poly A tail here and the 5' cap up here, and here's where it's going to join up with the ribosomes consisting of these two units. We'll get into this a little bit later on. The other thing I wanted to mention and remind you is that tRNA, that's transfer RNA, and rRNA, that's ribosomal RNA, they're produced in much the same way that mRNA is. We don't show them being produced here, but they have the same process. They're made in the nucleus, and they also move out into the cytoplasm. So the next thing we're going to talk about is the genetic code and how the code actually works. After we understand the code, we'll get into the actual translation process, which is a bit more complicated than what we've studied so far. So let's just go on with this. The genetic code. So once it's transcribed, the RNA can then be translated into proteins. And so we need to understand how to read this code. So we keep talking about genetic code. What does that actually mean? Now you're probably going to remember some of this from biology. You've certainly learned it, and it may come back to you as we go through it. So let's recall for a minute that proteins are made of these polypeptide chains of amino acids. So long chains of amino acids called polypeptides, poly meaning many. And it's really the order of those amino acids that determines the structure. And as we talked about, structure also determines function of the protein. So it's the order of those polypeptides is determined by the sequence of mRNA in the gene. So the mRNA contains the code for assembling the amino acids in this order, and that will result in a protein. So how does it that only 4 letters, so remember we only have A, C, G, and U in RNA, but we have 20 different amino acids as we mentioned. So how is it that 4 letters represent 20 amino acids? Well, what happens is each group of 3 mRNA bases makes up what we call a codon. So if you had a sequence AUG, we're going to call that a codon. And also the sequence UCU is a codon, GAU is a codon, CCG is a codon, AUC is a codon. Any combination of 3 of the 4 letters that you can make represents a codon. So this sequence here represents 5 codons. We have AUG here, UCU here, GAU here, CCG here, and AUC here. Those represent 5 codons. And then each codon codes for a single amino acid. So AUG represents the code for methionine. That's misspelled, sorry. And UCU codes for isoleucine. So how can we know what the codes are? Well, we use a handy thing called a codon wheel. So this wheel translates the various codons into amino acids. And the way it works is these large letters represent the first letter of the codon, and the medium letters represent the second letter, and the smallest represent the last letter. Okay? So we can see here we've got all our amino acids out here, and we have a code here. So let's go back here for a second. So if I wanted to know what GCA coded for, I could say G is my first letter, C is my second letter, and A, that's going to code for alanine. Or AUG codes for A, U here, G codes for methionine. So you can find a code for any of the amino acids using this device. Now there are other translating tables that you can use, but this is the one that I've been using. So how many combinations can you make given 4 letters taken 3 ways? Let's go back to math. We learned combinations and permutations. Think about it for a minute. Okay, if you've got 64, you would be correct. It's 4 to the 3rd power. So I can take, if I take 4 things 3 ways, I have 64 different combinations. And that's how many, theoretically, how many amino acids you could code for. But as we know, there's only 20 amino acids. So several of the codons can code for the same amino acid. Now why is it that 2 letters can't be sufficient? Well, 4 letters taken 2 ways is only 16. So that would not be enough combinations. You have to have more combinations. Okay, there's a couple of other things I need to point out here. All polypeptides, which are ultimately proteins, start with the sequence AUG. That's the RNA sequence that they all start with, and that codes for methionine. So if we look over here, AUG, that is our methionine. Now there's a couple of other codons here we need to look at. There's something called a stop codon. Again, let's go back. There's a stop codon, a stop codon. These don't code for amino acids at all. They just tell the ribosome when it's time to stop. It's really basically saying this is the end of the gene. Stop translating at this point. So you see UAA is a stop codon, and that's the purpose of those. Now, so here's our wheel. We'll use this in a minute. What I'd like you to try here is to go through the process. I'll try this at home, and we'll see what you came up with. Now, this is a coding strand of DNA, right? Now, remember, transcription starts with the template strand. So this is a coding strand. So if we're going to transcribe this into RNA, we need to find the template strand. In order to do that, we use our rules of base pairing. And so we need to come up with the template strand here using those rules. So A would be T, A, C, G, G, T, C, and so forth. So you can do that, and you can find the template strand. Now, if we want to take it a step further and transcribe that template, we would use the rules of base pairing again. But remember that RNA has no T. RNA only has U. So we can try that. And then you can divide it up into codons and use your codon wheel to see if you can translate this mRNA, the mRNA that results, into a protein. So I'd like you to try this. Come in and see what you're able to do. We'll go through it together. If you can't do it, that's fine, but I'd really like to see your attempts at this. Okay? And we'll pick it up again tomorrow.